All right, you've heard a lot about why we're here, right? We are charged, we are responsible with coming up with a fraction of the bioproducts, the feedstocks that are going to feed into the bioproducts that are part of the plan for energy globally. And that's a big responsibility. We need to develop biomass crops that will help carbon dioxide capture and storage while providing bioproducts and biofuels. This is going to require land. We want these feedstocks to be economically viable, environmentally sustainable, and we want to do so without increasing harm to our planet. And so we've got to think, how are we going to do this? As we think about how we're going to address these questions, I want to talk a little bit about the role that plant-microbe interactions can play. Now, plants, like all higher organisms like us, evolved with microorganisms, and in many cases forming beneficial partnerships or interactions, only some of which are known, actually very little of which is known. So this supportive soil microbiome is important for low input biofuel production. And that's important because we're going to be growing these crops likely on marginal lands where we're going to have to think about low inputs. Soils can contain large amounts of microbial biomass. Most of these microbial members, and we call these taxa, currently remain undescribed and have physiological and ecological attributes that are described here. So they're interacting with each other, with biology, they're interacting with the environment, and they're also interacting with the soil. Lots of these are very much unexplored, which is why it's a fun field, but also a very challenging field. These taxa don't work alone. They make up microbial communities that are highly diverse, in part because soil is highly varied. It's very heterogeneous. On the right here, I challenged my team to talk about how many microbes were in a single gram of soil. So we have sugar packets on our tables, maybe. We will later, maybe for teas, but that's about a gram of soil, a sugar packet of soil. There are millions of bacteria in there. But in the context of this conference room, I estimate there are 300 million species that could fit in here. So if it's a volume of a soil and there's lots of aggregates, that's 300 million exhibits that you could visit in a zoo to talk about who's in there. So it's in these soils and in this diversity, we know that microbial communities can have an important direct or indirect effect on plants or vice versa. So these effects on plants are driven by soil microbes and their role in moving gases and nutrients in the soil. And you'll note here that these are related to processes like greenhouse gases and emissions, carbon sequestration, and nitrogen availability. So this figure is nice. It shows the vertical arrows are microbial processes that interact with the soil atmospheric interface. And the curved arrows indicate some of the key processes that can occur within the soils that can regulate things like soil acidity, the availability of nitrogen, phosphorus, or nutrients, and the ease or the consumption of soil organic carbon pools. What this figure probably oversimplifies are the interrelated nature of these processes and the impacts of plants and the environment on these processes. So some of the key environments that we think about, especially in CABBI, between the microbes and the plants are perhaps manipulating or taking advantage of the interactions in response to things like drought, salinity, or extreme temperatures. And these are conditions we might see in marginal lands. So with their soil microbiomes, plants can be helped to respond to these more stressful growth environments. So in CABBI, I suggest that we are tackling 3 broad opportunities of how plant microbial interactions can help support the growth of our feedstocks. So the first is identifying the benefits microbes can provide to plants or soil health. So which taxa are providing what benefits and how. The second is using microbial indicators to inform or predict how nutrients, especially carbon and nitrogen, are moving through the agro-ecosystem. And the third is probably a blend between the two above, and that's a more systems context, and thinking not just about the plant, or not just about the soil, or not just about the microbes, but how does the whole system work together to respond to different stresses, and how can we manage that system? So in thinking about this class and what I wanted to teach you, I wanted to select a few, I think, foundational case studies of plant microbe interactions that I wanted you to know about. And the first example is a very famous bacteria called Agrobacterium tumefaciens. So over a century ago, this was noticed that a very unusual plant disease was observed in wooden plants, causing these tumor growths, and it's now identified as crown gall disease. And when these tumors were being studied, a few key observations came up. First, a bacteria was causing them. But second, interestingly, when you remove that bacterium, the plant would still have the disease. This disease would persist. So the key to how this occurs was unraveled with the development of molecular genetics. And there's a genetic structure called a plasmid, and it's where the genetic information is stored outside of the chromosome and in a bacteria. And that plasmid was moved from the bacteria and inserted into the plant host cell. And it took that information of how to grow tumors from the bacteria and put it into the plant cell. And this might be a little familiar to you because this was the pioneer of genetic modification of plants. In fact, in this natural sense of how to make tumors, that information being put into the cells, now we can decide what gets put into the plant cells. And so this bacterium has been used, for example, to insert genetic code that protects against insecticidal activity. And so this is a great example of one of the first things that showed direct plant-microbe interactions. The next example I wanted to explore together is the impact of bacteria living in partnership with plants or having a symbiotic association with plants. And I want to tell you a bit about Rhizobium, which is a genus of soil bacteria that fixes nitrogen. So as many of you know, nitrogen is one of the macronutrients required for plant growth and one of the most important elements for plants. It's also very abundant in the air. About 80% of what we breathe is nitrogen. But plants can't use that nitrogen. It needs to be in a different form in order for plants to use it. However, plants who associate with nitrogen-fixing bacteria, like Rhizobium, can convert atmospheric nitrogen into ammonia, a form that's more readily used by the plants. So a successful mutualistic relationship between plants and Rhizobium has been formed called biological nitrogen fixation. And what happens, which is very neat to me, is when the bacteria are present, the plant exudes enzymes from its roots that grow these little nodules that you see in these pictures, these little puffs, I guess. And then that provides a habitat that the bacteria can live in. The bacteria fix nitrogen, and then the plant's happy. As alluded to earlier, another plant function that can be impacted by bacteria is its response to drought. So water affects microbial function in a few different ways. It's a resource, it's a solvent, and it's also a transport medium, and each is important in regulating microbial function. And in this particular study, what happened is they compared drought-stressed beet, tobacco, and a perennial called goosefoot, and they looked at what happened when you inoculated it with a microorganism or a virus here. And you can see the control is super sad, super droughty, and when you inoculate, you can see a delayed drought response. Bringing this more central to some of our CABBI feedstocks, here's another study that's very exciting, and it looked at sorghum, and it looked at the timing of this response. And so we know that drought, the duration of the drought, and the timing of the drought can affect the negative impact it has on plants. And the question here was, well, how does it impact the microbiome? And so this particular group studied sorghum that was watered weekly for about 18 weeks, and then it simulated 2 drought conditions, one for 8 weeks, drought conditions, no watering, then followed by watering. And then it did a post-flowering drought where it flipped that. And so they did not water it, or they did water it at the beginning, and they simulated drought conditions after. And the question is, well, what happens to the microbiome during this time? So what I'm going to be showing you is a fingerprint, and that's what we do. We can do well, is fingerprint who is there. And so it's going to be a fingerprint of the community at each of these weeks. And the first thing I'm going to show you is the control. So these are the plants that were watered every week, the sorghum that was watered every week. And I'm going to show you 3 different compartments of the soil. So when I say soil, that can mean a big thing. So on the top here, we're talking about the bulk soil. This is generally, if you go dig a hole in the soil, what you're going to get. The second one is the rhizosphere soil. This is the soil attached to the roots of a plant. And the third is within the roots of the plant. So these are the different microbial communities in those 3 different soils. And what you see is in the roots, it's recruiting from the bulk soil. And this initial dynamic recruitment is followed by a stable period of who's there. So each of these colors is a color of a taxa that is there. And then you can see as it changes over time, the fingerprint is changing. Okay, let's look at the drought conditions. And this is where you had pre-flowering drought conditions. And you can see that it's pretty similar to the control conditions in the bulk soil, but if you look at the rhizosphere, what you see happening is you see this enrichment of these red and blue bars, which are actually actinobacteria and firmicutes. And so what's happening is these drought conditions are selecting for a different set of bacteria than watered soils. But you see that after the water comes back at week 8, it starts to look pretty stable, just like the control conditions. So what they did was they isolated these bacteria, and they put them on germ-free sorghum seedlings. and they wanted to see if it would affect their response to drought. In control soils, it really didn't make a difference, as you can see in the top panel. But on the bottom panel, in drought roots and shoots, you saw a modest but significant increase, at least in the roots growth, by inoculating by these actinobacteria and firmicutes. So what are the most exciting plant microbe questions, at least from my perspective, that we can tackle as CABBI, or what are the challenges maybe that we're going to have to be tackling to make our goals successful in this context? First is, how does stress, such as climate change or nutrient availability, influence these plant microbe interactions? Second, how can we manage these interactions? How can we work with the microbe-microbe interactions? How can we make management decisions to manipulate the benefits that we want to see in our feedstocks? And the third maybe speaks to the complexity of the system. We do a lot of scales of research. Wendy Yang was talking about how we talked about different scales of thinking in CABBI. We need to translate basic research, mechanistic understanding, to things like planting crops across the landscape, and that'll be one of the bigger challenges in how we translate plant microbe interactions into emerging crop plants. All right, thank you.